So we're going to talk about biologics, and you had a very thick morning of basic science, great talks about that, and we're going to talk more clinical talk, but though we started out with kind of an interesting thought piece on biologics, and that is who gets them, who among our patients gets biologics. So this was a study that was presented at EULAR this past year, and it's from Norway, which is very important because of the health system that they have. So it's a wealthy country. It's so wealthy it's not in the European Union because they have all that money from oil and gas. And as a result, one of the things that they do is they have a very high standard of living and also a very high standard of health care. All biologic DMARDs that they approve, we can talk about the process for that later, but the use of biologics in this country is higher than it is in almost all of the countries in Europe. And what they did was they looked at the people who were starting their first biologic and looked at the characteristics and to say how fast is it that people get started on treatment. So this is kind of the idea of treat to target, and we want to be seeing patients frequently and changing therapies in order to achieve the desired clinical target. And what it turned out with is correcting for a lot of things like disease activity, geographic locale, other features that might have had some relevance, that education was a very important for patients getting access to biologics. So I think fascinating because we always talk about in the US about inequality of access to medical care. A lot of that boils down to just pure economics. We also talk about race issues in the U.S., and here that variable is largely gone because they're much more of a homogeneous society. So in a place where those two variables are removed, education still emerges as a factor. The greater education you have, the more likely you are to get on a biologic agent faster. And I think that's I don't know that we know why that is. Is it that the doctors identify more with the educated patients? Is it that the patients are pushing it more? We don't know, but I think it's important because it being done in Norway, it takes out some of the variables that we always talk about here in the U.S.
I think if you can't pronounce Ustekinumab, you can't get on Ustekinumab. So that's where the education comes in. But this really is hard to explain, that smarter patients seek care more or are treated differently. It's really, my new best theory, which sort of steals from this morning's talk, is that there's a difference here maybe in microbiome, that these issues, the issue of socioeconomic status affecting how well people will respond or won't respond could also revert back to microbiome as one explanation. I really struggle to understand it outside of that one answer. Do you have another reason why this would be important?
No, but I think it fits with what Ted Pincus has been saying for a long time about how important education is. Of course, all his work is in the U.S. where these other factors enter in, but having a college diploma in some ways helps your healthcare. Of course, being a crazy liberal like he is, his solution is just to hand out everybody a college diploma,
or a reprint,
of a college, or of his work, or a college diploma. No, I think it's intriguing. Maybe it is something like the microbiome. So this, we put this in there. It's
wake you up.
I guess I've been in California way too long. You get the whole yin-yang thing going here. The point of this, though, is that we've learned, when we think of biologics, we've learned so much from our studies of and development in and use in the clinic of TNF inhibitors, but that's certainly not all that there is, and the success that we've seen has driven additional interest and many additional targets. And we have now, it's very exciting because we have some data that supports that it's not all about TNF all the time, that there are other ways to think of the immune response and maybe just continue to refine and modulate it in a way that might get immune regulation better. And then you see sort of the complexity of this, as we heard earlier this morning about the different subtypes of cells. And of course, the more we learn, hopefully the better we'll be able to pick targets to really change the immune system. And I think, as we're going to see, this may vary from disease to disease.
I think that we should have followed this with the Schett slide about the bench to bedside, the bedside to bench, because we've always worked on these targets, if you will, in choosing our therapies and the rationale. But in the end, we've learned more about how when you use these targets, you learn more about the pathogenesis of the disorder, and we'll see some of that with the results we're going to look at.
And so speaking of that, some of the, to take Lenny's charge and talk about some of the more interesting and exciting data in biologics across diseases, and not focus just on rheumatoid arthritis, as we often do in rheumatology. Very exciting data from last year's the ACR meeting, and that was on the IL-17 inhibitor, secukinumab. And here you see the data. I think an audience such as this is familiar with the data, but just to see a very clear separation between the actively treated patients with secukinumab and placebo with the ACR20 response. Of course, now part and parcel of our evaluation of psoriatic arthritis includes these other important manifestations like dactylitis and enthesitis. So nice, very stark, very clean data showing a good target, the persistence of a response over time. And of course, with this mechanism of action, a benefit is that from a clinical trial standpoint, you have kind of a built-in positive control, and that is that you have the skin responses. And here you see, as you would have expected based on the earlier data from skin psoriasis studies, great PASI 75 responses. In fact, that's why our colleagues have been talking about PASI 90 and 100 and such. You also see here is an effect on x-rays, which I think is really, it's very interesting because it's gotten so hard, hasn't it, in rheumatoid arthritis to show an x-ray benefit. You have better treatment overall, you have less x-ray progression overall, so you have a shorter period of time during which you can expose active patients to a placebo treatment and not rescue them. Given all those caveats, and in psoriatic arthritis, it's even tougher than rheumatoid to show a positive effect. But what you see here, even though the magnitude of the change is small, it's 0.6 of a sharp unit, but quite significantly different. So this mechanism of action can lead to improvement in x-rays.
So the interesting thing, I think, so far, again, getting back from that bedside to bench lessons, the effects on dactylitis and enthesitis, but we also know from other studies not published here is that it doesn't work for uveitis. And there's also the story of exacerbation when you use secukinumab and other IL-17 inhibitors of exacerbation of Crohn's disease. So again, we tend to think of those as extra-articular manifestations of disease, but they're not all the same mechanism that's underlying them, and I think that this drug sort of highlights that. I think the safety on this drug seems really acceptable, to say the least. I think from an animal model, you might have expected fungal infections, deep tissue fungal infections, but really we have basically these minor thrush cases occurring in very low numbers, so that's also a good story. I think that the challenge thus far is how you use the drug. Are you going to use the IV loading dose, which is used in the future study, followed by monthly sub-Q, or are you going to use the just the sub-Q regimen as they do in dermatology with psoriasis?
Yeah, I think they're great points, especially the bowel, because as we know, if you take asymptomatic psoriatic arthritis patients and do colonoscopy and biopsy, you find probably a third of them have colonic inflammation that is basically Crohn's disease, and that number is even higher in ankylosing spondylitis patients. So because this mechanism of action does not seem to be useful in Crohn's disease anyway, are we going to see anything related to that? So it would be something that we'll certainly want to see data about. And as you mentioned, the sub-Q is probably going to be the way we're going to do in the clinic. Where are we going to use therapies like this? And I think this is, it's always a challenge. We always want to know what a therapy is going to do in the group of people for whom we've tried our other therapies. So the psoriatic arthritis, the TNF exposed patients. People came to you, you maybe gave them a DMARD, maybe not, got on a TNF inhibitor. Maybe they had some response, maybe very little, wasn't good enough. You put them on a study, what do you see? And this is sort of a mixed bag, because you see that there is a little bit of a cut. There is a little bit of a hit in this study with a little bit less response among patients who had not been on a TNF inhibitor before. It's interesting because it looks like it may have something to do with dose. If you look at the bottom two graphs, you see on the left-hand side that in the TNF-naive population, it doesn't look like 300 gets you anything over 150. So why not go with the 150? But if you look on the right-hand graph, you say, well, it looks like in the TNF-exposed population, the higher dose does do better. So I think we're going to have to learn about this medication and other medicines in this class, how best to use it. Do you start it high and taper it? Do you dose it based not only on the PNF exposure? Are there other factors? So exciting data. We have to learn about it.
So again, the top study, Future 1, there's an IV loading dose, 10 milligrams per kilogram for the first 3 shots, and then they get monthly injections. The bottom is just sub-Q doses right away. And again, the sub-Q is a recommendation per package insert when this drug is currently available for patients with psoriasis. So the same actually was done in ankylosing spondylitis. They have a Measure 1 and a Measure 2 study. The Measure 1, there's an IV loading dose. Here, the ankylosing spondylitis here, the outcome is 16 weeks as opposed to 24 weeks. And again, you're seeing very good I think numbers that you compare to anything else out there right now as far as these ASAS-20 responses and ASAS-40 responses in both the Measure 1 and Measure 2. And here you're seeing, again, patients who are TNF experienced versus those who are TNF-naïve. A bit of a drop with the TNF incomplete responders, as you might expect, as we've seen with other drugs in rheumatoid arthritis. In the Measure 2, where they just get sub-Q, again, the same drop occurs, although there seems to be some benefit, I think, in the IV loading. I'm not sure in the long run that it would stand up, but in these short-term trials, it looks like there might be a benefit. So it'll be interesting to see when it comes around to how the drug is promoted, or what its dosing is going to be when and if it's approved for these conditions, psoriatic arthritis and ankylosing spondylitis.
Yeah, and in AS, I think you could say there's maybe a bigger unmet need, because certainly if you're focusing on axial disease, there's no benefit to DMARDs. So you have non-steroidals, then you have your TNF inhibitors. It's good to have other choices. This data I think is really exciting in that regard. Some of the data we saw with ustekinumab, and again, like you said, the bench to bedside, we're sort of learning about the pathophysiology of the disease backwards, but it's certainly not just a theoretic. This is great to have because we have these people in the clinic, and the more options, the better if we really want to get them to be doing well.
So next is, again, ankylosing spondylitis, but now actually we're going to look at the non-radiographic axial SPA, which is sort of a hot-button issue. Artie, what do you call these other versions of SPA that are out there?
Well, I think it's the ASAS people. It's kind of a political group. So uveitis is iritis is uveal spondyloarthropathy. Crohn's disease is colonic spondyloarthropathy. Everything is a form of spondyloarthropathy if you're a part of ASAS.
So. Again, this is one of the reasons why this failed in front of the FDA. There was a lot of concern, at least in this country, about how appropriate this terminology is. But in this trial, these are patients who met criteria, who had back pain for less than 5 years, who had BASDAI scores of greater than 4, and had failed nonsteroidals. And in a placebo-controlled 16-week trial, they do well when they're put on golimumab compared to placebo, as shown on the left. But you can see one of the main issues that was discussed at the FDA advisory panel that met on this, that when you looked at patients who either had an abnormal MRI, basically reinforcing that they really do have a sacroiliitis, or those who had an elevated CRP at baseline, this is where you saw results very similar to what you see in the overall results. But if you look at a subset that doesn't have the MRI or the CRP, using this definition fails, and you really can't make a good case for using a TNF inhibitor in such patients. So again, one of the things that didn't happen but might happen in the future is that if this becomes an indication, it'll probably come with the hook that you must have a CRP and or an abnormal MRI to get the drug.
So in that last group, is that just bacomyalgia?
There was a lot of discussion about can rheumatologists and other doctors tell a difference between fibromyalgia and non-radiographic axial SPA that doesn't have a CRP or an MRI. I don't think we have a problem with that, but there was a lot of discussion on that issue.
And we do studies, and we are doing a study now. And I guess doing studies, it's kind of like hunting. During duck season, you see nothing but deer. They're coming around and kicking the butt. You look for things, but I swear these people do not exist. Anybody who really has low back pain, you get the X-ray, and they have changes already. And you don't really have to have a lot to get New York criteria.
They don't exist when you need them to
When you
enroll them in
need
a
them,
study.
exactly.
That's the problem. When that FDA hearing happened, I had 3 show up in my clinic that week, brand new, met the criteria and everything. So I agree. It's sort of a strange group, and whether or not we should be going after them, I don't know. Biologic use in scleroderma. I think this is obviously a gigantic challenge for any of us who manage patients. We're looking for some hope of a new therapy, and in this case, they're targeting IL-6 because of animal models which suggest that IL-6 expression is increased in an animal model that's similar to scleroderma. So they have an 87-patient trial with diffuse disease. They have less than 5 years of disease, an elevated acute phase reactant, and they either received Tocilizumab at 162 milligrams subcutaneously versus placebo. And their primary endpoint here is a modified Rodnan skin score, which I learned from my time at Pittsburgh, which is the easiest and lowest hurdle to reach in any scleroderma trial, albeit this is a controlled clinical trial. They did not meet their endpoint. Now, they did make a case for the placebo population having less meaningful reduction in the skin scores, 26% versus those on Tocilizumab at 43%, and that they did have what they thought were reasonably higher numbers for FVC change, improvement in FVCs. So again, some suggestion here, but in the world of scleroderma, this is yet another disappointment. I can't see this going forward. Although I'd like to see something go forward in scleroderma.
Yeah, a tremendous unmet need, but this study too, I think one of the important considerations about it is the study design. And this was the Goldilocks study. The patients had to have bad scleroderma, but not too bad. Had it for some years, but not too many years. It had to be just right. And screening patients, the hit rate was less than 10%. So you're weeding out a whole group of the potential patients with scleroderma who really want to be in a study, really want to do something. They're great patients, and you're focusing just on a small group. So I think the data is super interesting, but the fact that they didn't meet it in such a highly cherry-picked group of patients. Now, that cherry-picking might have worked against them. Who knew if they just took everybody who came that they might not have done better. So this gets to, I think, for study design, the more restrictive the study design, the smaller the group of people that you enroll, and then sometimes the data gets skewed. I don't know. I don't know if this is good. Is it worth pushing further?
No, but it is a good idea to get those just right patients when you're doing clinical trials. Sifalimumab, an anti-IFN-alpha inhibitor, in this phase 2 trial was reported by Kavanaugh at the ACR meeting. This is a 400-patient trial of those with active seropositive lupus who had active disease despite standard of care. No BILAG A or 2Bs, and no patients with active renal or CNS disease. And they either received one of several doses of Sifalimumab or placebo for a year, and their primary endpoint was the responder index at 1 year, and then they did an exploratory with another definition of response called the BICLA. The safety, there was probably not much more, in the way of safety differences between the two. 2 more deaths in the Sifalimumab group versus placebo, but overall those are very low numbers. They did have significantly more zoster in patients receiving the alpha interferon inhibitor. The zoster event rate was about 0.9% on placebo and ranged from 4 to almost 10% in patients who are receiving the alpha interferon, so 4 to maybe even 10 times higher. But on the right, you can see, again, what happened. We're looking at the responder index after 1 year. To me, it looks like a graph that's not that dissimilar to what we saw with Benlysta, where, again, you have fairly high placebo response rates, but at the highest dose, 1,200 milligrams, they did achieve significance, suggesting now that this might be yet another approvable drug. If they use more stringent criteria, not the BICLA, but what was called the SRI 6 and 8, they actually seem to have a little higher number of responders. And then using the BICLA, again, they only seem to approach response. It's thought that I think the BICLAs might be a little less sensitive a tool in measuring lupus responses. problem with most of these trials is that BICLA, SRI, SLEDI, whatever, these are not appropriate to practice, and I think it's hard for most of us to interpret these trials, what they really will mean to our patients, because certainly the tools don't in any way really approximate what goes on in real life.
Yeah, it looks very much like the belimumab data, and you wonder, what is it with these lupus studies? Do you need 1,000 patients to show if it is a significant difference? And I think your point is a great one about the instruments. Thank God we can write belimumab without some high school kid in the pre-auth office asking you what bilag A and B they have, because that could happen. These are all approved by these Rube Goldbergian complex outcomes, and that has absolutely no translation into clinical practice. And I wonder if it's actually, if they wouldn't do better, what if you just stick to the patient-reported outcomes? How you doing? Are you doing better than, maybe, the Brooklyn Hack. Maybe we're losing therapies that could be effective. This study, it sure does look like the belimumab data. No clear dose effect, a very small effect size, even in a large study. So, I don't know.
87% had an alpha interferon signal here. So maybe they were a homogeneous population on the basis of that, but only about 25% with double-stranded DNA positive. Even though we had responses here, there was no significant change in complement levels or double-stranded DNA levels. So again, this is, to me, I thought alpha interferon inhibition, based on a lot of the work that's been running around in the last 5 years, would have suggested that this really would work, but it's not, I think, lived up to its expectation. So pregnancy, we had a few abstracts on pregnancy this year. We covered this at the ACR. Otis actually had a nice abstract on its experience with adalimumab. It's a lot of work to get this data. It's a lot of work to keep patients in a trial where you're all throughout the pregnancy and then postpartum and watching the kid and counting the kid's toes and fingers and these sort of things. So their numbers are never very impressive, but we can count them later. They did have 74 women exposed to adalimumab from the first trimester on, and interestingly, if you'll see here, is that 43% continued adalimumab throughout the pregnancy, and they actually have matched controls here, RA patients not pregnant and non-RA patients, and when you look across the board, live births are not different, spontaneous fetal loss, again, not loss, that's a difference, and major malformations, not any difference. And again, the historic background for malformations in this population is going to be somewhere between 3 and 6% for the most part, and that seems to fall within that range. Abatacept had an abstract also at the meeting detailing their experience in a number of pregnancies between 95 and 2014. The numbers are small, but again, no abnormalities outside of that expected, a very low number of congenital anomalies. So again, adalimumab, a class B pregnancy risk, and abatacept, a class C pregnancy risk. Nothing surprising there. Whether or not you would jump in and want to use that based on this data, I don't think you would, but there is, I think, a growing body of evidence about at least the TNF inhibitors that is repeatedly showing no new surprises and whatnot. I don't think we have enough data on the class C drugs, the Tocilizumab, abatacept, rituximab, to know what their use may be in and around pregnancy.
How many, let's see, a show of hands, how many people have prescribed or encouraged a patient with rheumatoid arthritis or other immune diseases to stay on a biologic agent, a TNF inhibitor? How many?
During pregnancy.
So some people are raising their hands like this, like. But a lot of hands are going up, and I think
we're all gastroenterologists who have a much greater experience with this than we do. So but that's surprising. That's actually, my estimate was always about maybe 20%. And of rheumatologists that actually would be more inclined to use TNF inhibitors, surveys show that at least a third of your patients will continue those drugs during pregnancy or resurrect them during pregnancy. So we talked about class B and class C pregnancy risk. This is all changed as of December 2014. Artie?
Yeah, so how many people know that the pregnancy labeling is changing? Wow. Okay. This is big stuff because Jack said, we talk ABCDX, don't we? We all learned it. We kind of tell the patients as if they're supposed to know, well, that drug is a B, and they're like, and there's no A's, and there's problems with the system. So the ABCDX is established in '79. And it was great, but it also had a lot of problems. There's no A's, because A means that there are well-controlled studies in pregnant women, and of course the FDA rules essentially prohibit studies on women, to conduct on pregnant women. To conduct a study on a pregnant woman, the threshold is incredibly high. You'd have to be nuts to try to do it, because you basically have to ensure that there's no bad outcome for the fetus, and that there's going to be a good outcome for both the mother and the fetus. So there's no A's. Also, a lot of the things don't make sense. X means, definitely don't do it in the case of methotrexate, but it also means you don't need to do it because it's not an issue like the statins. Statins are X's because you shouldn't be using a statin in pregnancy. You don't have to. It's not that they cause severe problems. The hydroxychloroquine is a C, even though most of us approach our patients and say it's very safe. So there's a lot of problems with it, and the difficulty from the agency standpoint is that people are making bad decisions and perhaps terminating pregnancies unnecessarily when that is done. So what they did, and this has been it's like all good federal things, this has been the work for about 9 years that they've been doing this, but it actually just happened, and it's amazing that that this hasn't hit us, because it's going to hit us very soon. The letters are gone for new drugs. So as of June, new drugs approved after June, there's going to be don't spend your time looking for the ABCDX, it will be gone. What they will have is a narrative. So the narrative will have, just like the discussion Jack just led about the adalimumab, that will be put in a narrative form, and it will say a summary, show the data that there is, or say that there is no data. A lot of drugs, there is no data. Point you to registries, to things like Otis or Mother to Baby or others for our Canadian colleagues, and also include information about leaving the disease that it's approved for untreated. That's a big issue with our GI colleagues. Crohn's, the GI people are probably way ahead of us in this, and that they realize that the most important thing for fetal health is mother health. And if the mother's sick, the fetus is starting behind the 8 ball, and they're more aggressive in trying to control the disease than we are. So I don't know. Jack, you look forward to this? You worry about this? What do you think of the change in the rule?
Well, I remember back in January of 2014, you ran the ACR pregnancy summit that we had that became a publication that came out last month, and the ACR is going to have guidelines on pregnancy and its management. But at that summit, we had all the stakeholders there, the companies, the FDA was there, a lot of concerned investigators. The FDA actually did this presentation and was pretty proud of their presentation, but they really took it on the chin from all these experts in the audience who were saying, are you kidding me? You're getting rid of the simple ABCDX, and now you're going to replace it with 19 pages of print and studies that no one's ever going to read. It's a gigantic step backwards in patient management. So, the best label might be the one that says, as you said, no studies available. Sorry, Charlie. There's a registry. Call these people. That might be the best. But it's really also the least revealing. So yes, they're going to put in detail, and it's going to be detail about maternal issues, about fetal issues, about registries, about lactation, and there'll be some specific verbiage in there that's drawn from studies the company may have done or what's appeared in the literature, but it's going to be lengthy and it's going to be detailed. And again, as a group, physicians are not very good at reading the product insert or the package label. We tend to go to it to find that one answer we need to get on with patient care. We want to do it in a fast, expedient manner. That's why ABCDX was so great. This is going to slow us down. This is going to frustrate a lot of people. But as already said, I think you have to get ready. It's happened.
Yeah, I think it's, as rheumatologists, we'll know what to say for methotrexate, for leflunomide, for the TNF inhibitors. The part that I don't like about it is that when you have the patient who you already said we're going to continue the adalimumab, and they say, hey doc, what about this drug? And then you're on the electronic record anyway, so you start Wikipediaing that drug, and you don't even know what the drug is, are you going to read 3 pages of stuff to say, geez, I don't know, just stop it. I can't even spell this drug, and I don't know what it is. So I think in some ways we will miss the system, but there were problems with the system.
I got a text this morning from a dermatologist in Dallas who said to me, can you tell me what to do? My OB wants me to send the patient to a genetic counselor if she's going to receive Enbrel around the time of pregnancy. So now the OB, who doesn't really understand the situation, is sending the patient to a genetic counselor who definitely doesn't understand the situation, right? And they want to know what we think in rheumatology. Well, what we think in rheumatology is communication between the disciplines is absolutely horrible, and that's why we need guidelines and better education on this.
All right, so other things in the use of biologics. So there's an interesting presentation that was done at EULAR from Japan, from Tom Takeuchi, and it looked at whether or not, or how people did with loading. I personally, I don't think loading adds that much, but you think about our drugs, we load infliximab, because that's the way the studies were done. Our GI colleagues and our dermatology colleagues load most of their TNF inhibitors, so they give higher doses and then cut back. I don't think, we tend not to. And for a drug like was studied here, Tocilizumab, for me, that's 2 prior authorizations. So I would just assume start them on a drug because I think, now they'll catch up. And this is interesting data. 2 studies from Japan basically differed in whether or not they used the loading dose or didn't use the loading dose and just went to the therapeutic dose straight away. And this is data I think you can debate. I think that it shows that if you load, you do better faster than if you don't load in both studies. And do they catch up at the end? Well, kind of, but if you're an area under the curve kind of person, it looks like loading is a little bit better, and it's not that much more of a deal in terms of the safety, cost, other aspects. I think they do better, and this has caused me to reevaluate my otherwise antipathy toward loading, to say, just start it and you'll catch up fast. But Jack, you disagree.
Yeah, and I also don't have an infusion center, so I would think that may color my thinking on this, but I don't see, again, from an area under the curve and where you get to, and even the magnitude of benefit at the outset, especially in JRapid, you're comparing orange dots to red dots to blue dots being ACR 20, 50, 70s. Rapid really shows very little at that week 4 time point, a little better at week 8. The Hikari is a little bit better at week 4 and not so good at week 8. So it's inconsistent. And then certainly out of week 52 and 40, it's really not any different. So I can't see, this would be good evidence for me not to use any loading in my patients.
Didn't JRapid open for 50 Cent here in Cleveland not so long ago? I think wasn't that the concert? Other ways to, tough audience. I got a laser, I'm not afraid to use it. Other ways to optimize therapy, and again, with a shout out to our GI colleagues, drug levels. So the GI people in our institution have been so strong advocates of this that now we can actually get the test done. I don't know how it is in your universities or your hospital settings, but you send a sample to the lab, and they don't know what it is, they just throw it out, sample bad. But the GI people have really pushed on them, and now we can get drug levels which have to be sent out. Do we need them? Do we use them? The GI colleagues and gastroenterologists here, they think you get a lot of good information. Of course, Crohn's disease can have a protein-losing enteropathy, so you could be losing biologic product out the gut, where we don't have that particular issue, but we certainly both wrestle with the ideas of immunogenicity. This came out as a suggestion published in ARD a year and a half ago, saying where you might use these. And, I don't know. I think it kind of makes sense. Somebody isn't doing well, you measure the drug. And if they have good drug concentration, then maybe you switch. If they have low drug concentration, well, this says increased dose or frequency, but of course in real life, if they have low drug concentration, you say, Mrs. Smith, how often are you taking your etanercept? Really? I mean, really? Then, and the antibodies, whether or not they have a role in this, I don't know. I don't think anybody. How many people or how many people have gotten a drug level? Just a few. Okay. I don't know. I think could there be for a role for this?
It makes great sense, and why we don't do it, I think we've never been persuaded that all the buzz about anti-drug antibodies, specifically with adalimumab, have really amounted to enough data that should change the way we actually use that drug. And I think that's guiding a lot of our principles here. But with this construct, this makes great sense. Unfortunately, this has never been applied to one of our populations to show that this actually yields better outcomes. It has been applied in GI with better outcomes. So again, whether we're going to follow their lead remains to be seen. Lenny.
I have a shout out here. I've got infliximab levels, but how many other drugs are commercially available?
Adalimumab and infliximab are widely available. In Europe, I think you can also get golimumab.
So again, remains to be seen, but I wouldn't be surprised to see trials designed on this sort of approach. I mean, and maybe that's the way to go. Maybe we'd like to. These are great drugs. We like the way they work, but the fact is, they still don't work in 30 or 40% of our patients. If there was a smarter way that could get us to higher outcomes, then we should be doing it. And that's the whole idea behind escalating therapy, de-escalating therapy, or even using biomarkers to guide therapy. That's sort of what we think our future is.
So the next topic we'll go through, Jonathan Kay is going to cover this in much greater detail, but certainly relevant to the use of biologics, and that is, can you taper them or potentially discontinue? And of course, our history and tradition has been no, because the earliest days, this is one of the first studies Larry Moreland published for etanercept way back when, and when you stop therapy, people flare. And these people, like some of the patients we still see, had the absolutely worst, toughest rheumatoid arthritis. Why would you stop it when they were doing well? And the only reason to would be if they can continue to do well. A number of studies address this. One is the BEST study, which is out 10 years now, and you're very familiar with this. You know the design. Two interesting bits of data that were presented relatively recently for this. If you look in the middle of the box, number of people in drug-free remission is similar across groups. Kind of makes sense. Treat to target, escalate therapy, do well, you can back off therapy. About 15% of your patients, early RA, treated aggressively, can come off all drugs. And that's, I think, a useful number for us in the clinic. You see the bullet down the bottom, very interesting in that the ACPA negative patients could do this much more successfully. So 15% overall. If you're ACPA negative, that's 27%. If you're ACPA positive, 6%. And I think that's actually remarkable. That's a really strong biomarker, considering that the way the study was designed should have obviated a lot of the impact on ACPA positivity or negativity, because you're selecting people by disease activity. And then the PRIZE study, which I think highlighted two fascinating aspects of the whole idea of tapering or discontinuing. One is the better you do, the better you will do. And you see that on the right-hand graph. People who in the PRIZE study, early rheumatoid arthritis, treated aggressively, those who were in super, super deep remission, DAS 28 less than 1.5, were able to stay in remission much more effectively than those who were in remission, but not quite as deep. And then the other aspect of it is the people who responded faster. So we all see these patients in our clinic. The one patient, you put them on a TNF inhibitor, they come back in the next visit, and you're the smartest doctor in the world. Why didn't we do this sooner? I feel great. I'm planting roses again. And then you have another patient who you put on a TNF inhibitor, and 2, 3, 4 months, they're like, I'm better. And then by 6 months, they're like, doc, I'm really a lot better. And it looks like, obviously, we want people to be the former, but if you look at the graph on the left-hand side, people who got into remission fast, much more likely to stay in remission when therapy was discontinued, way more effectively than those who got there slower. Do we stop therapies? Well, what therapies do we stop? This is a study that was presented at ACR from Georg Schett, presented it. They stopped the TNF inhibitor, they stopped the methotrexate, they stopped the prednisone, and like a lot of the data that you see in this regard, it depends on how you look at it. You could look at this and say, wow, half the people were able to come off all therapy. Or you could look at it and say, well, gosh, why do that? 35% of your people are going to flare. Why would you do that to them? A very interesting part is, in terms of study design, is that orange box, 16% of people, very deep, very long remission, flared even though they stayed on their drug. So that's what we all know when we see our patients. The disease waxes and wanes, which makes study hard. And then finally, Jack, back to the idea of the drug levels. What about this?
Well, this is more of a real practice observational design, where they took a cohort and the objective was to have one portion have a scheduled withdrawal design, and the others are going to have their routine care. So these are patients who are on either adalimumab or etanercept, and they just wanted to see if there was a significant difference between the two, hence the non-inferiority design. In the end, even though there was a group that had a scheduled reduction in dosing intervals trying to get off the drug, at the end of 18 months, discontinuation was seen only in 20%. An increase in the interval between dosing was seen in almost half the patients. No reduction was possible in over a third of the patients. So these investigators said in practice, this was actually not that hard. A significant number of people were able to either change their interval or get off drug, and that there was an increase in the Sharp score in the dose reduction group, but drug and drug antibody levels, or drug levels and anti-drug antibody levels were similar in both groups and did not predict those who could actually successfully discontinue or lower their TNF inhibitors. So again, it sort of argues that this is doable, but then there are other studies like the one you just showed that there's a price to pay that you do this. When I talk to rheumatologists about this, there's a group that says, look, I struggle so hard to get people to this state. It'd be crazy for me to buy into the patient's desire or someone's desire to reduce the dose or get them off the drug. Whereas there's others like myself who say, if the patient really wants to get there and they've got the numbers like the depth of remission or speed of remission that might allow them to do it, then why not go ahead and try it?
Yeah, and the two final points on this is it's not just TNF inhibitors. People are looking at this in other drugs. This was just published very recently, the AVERT study where they tried this with adalimumab, and you see at the far right of the graph, if you're in combo therapy, yeah, you did a little better in terms of coming off all treatment, but the numbers are still quite small, and it doesn't look like you did that much better. And then finally, to Jack's last point, another thing that was just published with Tocilizumab was that if you, and this is a big concern, if you stop people, they're doing great, they're in remission, you stop, well, then you put them back on treatment. You don't capture them right away. We've known this actually going way back to the Tenvoldi article from the early 90s in Lancet, that you take people in remission, stop their therapy, put them back on treatment, you don't get capture response immediately. Sometimes it goes out like it was in this study, and you see in the bottom right, it took them a while to get back to close to where they were. So there is a price to pay for discontinuing therapy. And there's just a mulligan, like Jack and I call it when we do this at ACR. This is a study that we published looking at the Corona database as the source of information, just saying that some people do this. And I think this is real doctors, real patients who just, both of them came to and said, let's stop therapy. How long do you go out? And you see here that at a year, 73% of people were still in low disease activity. Most people got a year and a half. Some people got 3 years. Unfortunately, we don't know who. We don't know which patients can do this. None of the factors were super strong. A quick response did do better, but we still don't know who can do this.
Well, the end of benefit, the higher CDAI's patient scores and HACCT discontinuation, those are things that you might look at. But I think the things that what I tell my patients, you show me a year of remission, and then we can have a discussion about reducing your drug. I don't know if that really applies. So based on these data. This is I think goes to the other issue of how often you really are in remission. This is a sort of small study, but it goes to that issue: is a clinical measure enough to say that someone's in remission, or how deep do you have to go to really define remission? They had 14 RA patients in remission, meeting the Pinal's criteria, meaning no swollen joints, no tender joints, no morning stiffness, no acute phase reactants,
no pain,
zero
no fatigue,
across the board.
Tough.
And yet they did synovial biopsies and ultrasound and MRI and showed that a high percentage of these patients had significant amounts of synovitis, either by biopsy or by grayscale synovitis or power Doppler. And so the idea here is that even when it's great, it's not that great, and so there's a lot of subclinical microscopic disease that's sort of should be an argument against wanting to get off of these drugs.
Yeah, I think when you go back and you read the Pinal's paper from 81, it's an absolute brilliant paper, and they said they wanted remission to be complete. They wanted no immunologic activity, and they realized, certainly at that time, but although still true, we can't go poking people's joints all the time or looking so invasively, so they settled on a clinical group of criteria that were very, very stringent. We all kind of forgot about them for many years because nobody was achieving them. But even if you do achieve that, you can still have immunologic activity. I guess we have to figure out what are the implications of that. So cost is an issue with biologics. We haven't said much about cost. It's, of course, the 800-pound gorilla in the room. That's why it involves every single patient decision that we make. It's interesting. I have a visiting scholar with me in San Diego from Canada, and as he's in the room, he's like, every single patient, cost is some aspect of what we talk about. It's crazy. But for the biologics, there is the expensive cost. The high cost. This was I thought a fascinating presentation. This was from Finland, and a benefit of that is their social system and their homogeneity to the population, which takes away some of the difficulties doing pharmacoeconomic analyses in a place like the U.S. Long story short, what they found is that at 2 years, it was about a wash, meaning the infliximab was more expensive, but the people who got infliximab went to work more often than those who didn't. So gets the idea of personalizing. If we knew who those people were, then you'd save all the costs of overtreating the people for whom you're not getting an additional benefit on top of the triple therapy, but we still don't know those people. But costs, incredibly important, isn't it? And here, you say, why are we talking about this? And for years, I think we as rheumatologists have been sort of under the radar a little bit. Very exciting developments in biotechnology and in new pharmaceuticals. We're not under anybody's radar anymore. This is from a trade journal, just saying where's all the money going to and what's going to be chopped. And there you see, there we are right there. The TNF inhibitors, top priority for reducing the costs. So how do we reduce the cost of these?
Biosimilars. So, Artie and I were working on biosimilar slides, and I said, well, we need a good picture, and I had a few, and I thought of putting Ed Keystone up there with Jay Keystone, the identical twins, because we might recognize at least both of them. But I found this one. Artie, anybody knows Artie knows he likes the Homer Simpson. And, yep, there you go. And he comes with a built-in soundtrack. But this is obviously a hot-button issue for a lot of us. This is a major way that cost is going to be contained in the future. This has become a major legislative issue around the country. And there's, there are people in favor of. There's the generic pharmaceutical association, imagine them being against it, are saying that that this is going to be a problem. So, but there's a lot of work going on in this area, and there was a lot that actually happened at the ACR.
Yeah, this too is going to be covered by Dr. Kay and Dr. Strand as well. So just go through this sort of quickly. Just a couple of things, a couple of the highlights of things that were at ACR. Tons of posters on this. One that was intriguing is that we know a lot, and we'll hear a lot more from Jonathan from Vibeke about the biosimilar infliximab, remsima, that's widely available in Europe. But there's now a biosimilar etanercept that's available in Korea, and it was approved at the ACR. In fact, the guy announced it from the podium. I thought he got that. That was pretty cool to be able to, oh, by the way, this drug is approved. Yo. He didn't really do that. Yo probably means something else in Korean. I don't know. But that was very cool. And then at the bottom, I think is incredibly important, and that is the biocopies or biomimics, or we don't know exactly what they're going to call. There was a really crappy abstract at ADCR, crappy meaning from a data standpoint. They just said they had a lot more reactions with the biomimic rituximab, which is called Tocilizumab, which is, what a horrible name for a drug, isn't it?
That's the greatest name. You kidding me? Tocilizumab.
That's what happens.
Built in the marketing campaign.
That's what happens to your GI tract when you go to Mexico to get it. But anyway, it was withdrawn from the market, and I think we'll hear in great detail about this from Jonathan and from Vibeke. But I don't know if the public is how clearly they're going to distinguish biocopies and biomimics, and will you have patients showing up at your door saying, "Hey doc, I got this chinfliximab, I got it online. Will you infuse it into me?" They may not know the subtle differences, so it's going to be something that happens.
You got to start developing that.
All right, so what about biomarkers? We've danced around biomarkers. Jack, what do we got for biomarkers?
This is a big push, but still, I think a lot to be done. Vectra, the MBDA is out there. They actually applied their tool to the SWIFOT trial, where they looked at the population of patients who were the early RA patients who were methotrexate non-responders, and then saw what happened to the Vectra score in the patients who were randomized to either triple therapy or the biologic therapy. And what they did show were you just look at the orange bars, the patients with the highest levels seem to have the best response to triple therapy. They also showed the patients who had the greatest, not shown on the slide, the greatest decline in the Vectra score were more likely to respond to triple therapy, and those with the less decline were more likely to respond to TNF inhibitor therapy. But if you look at the numbers for the green levels that are on the lower scores, it didn't seem to matter whether you got triple therapy or TNF inhibitor. So again, suggesting that maybe Vectra can distinguish which patient should get which drug, I think is one interpretation of this. I think it's a bit of a stretch myself, but I think we need more research in this area using this tool and others.
Yeah, I think an interesting use of this that they said, so it's kind of this tool, and they're experimenting with how to use it. One was clinical trials, and they said that if you accepted that the abnormal Vectra was a legitimate reason to be in the study in terms of, instead of the acute phase reactant, you get 25% more patients in studies, because now typically, because of bad investigators and bad studies, you have to have a high SED rate or CRP to get in a study. We all know we see lots of people who have abundant synovitis yet don't have a very high SED rate, so it might be a tool to put
so that just
more
got every
people.
investigator who wants to make more money excited. The FDA is never going to act on that, so it's a lot of nonsense talk.
Another thing with biomarkers, I think we have to sort of think outside the box a little bit. We tend, and our colleagues in oncology have had great success with a single home run. If you have certain polymorphisms in certain cell surface proteins or signaling molecules that are, of course, directly relevant to certain kinds of cancers, the prediction, if you have this, you have a 90% chance for a kinase inhibitor. If you don't have this defect, then you have a 0% chance. But that's like a single home run. We'll never have that. So I think we have to think more complicated. And this is analysis looking at that in one way. This looked at the ADActa study, which was adalimumab versus tocilizumab. And what you see is that if you have a pattern of, and this is all based on immunohistopathology of synovial biopsy specimens, CXCL13 expression is a surrogate for lymphoid aggregates within the synovium. The soluble ICAM was a surrogate for myeloid. There's also a lymphoid pattern. So what you see is the extremes. If you have a high lymphoid pattern and a low myeloid pattern, you had a really good response to tocilizumab, and the opposite for adalimumab. Now, of course, in the article, what you see is that there's the people at the outliers, they're probably 40%. The people in the middle, where you had high on both or low on both, then it's a real mixture. But I think we need to have a more complex approach to biomarkers, not just to have a single, maybe a group of individual ones that can be put together, but looked at in different ways.
We are not going to show, there's other ones out there. There are people looking at, including calprotectin. IL-33 is an interesting one, because in the bottom line, we're sort of pressed for time here. Bottom line is, if you take IL-33 and you combine it with other known predictors for Rituxan responses, on the right you see what happens if you're just looking at who's going to have a EULAR response. And if you combine just seropositivity with first IL-33, then with IgG levels, that when you combine all three of these together, that you actually can predict responsiveness to Rituxan up to 92% of patients. So, I mean, that's the kind of biomarker or set of tools one needs, I think, to march forward. So, again, I don't know if there's going to be more work in this particular area with IL-33, but it was, I think, quite compelling to just look at those three factors: seropositivity, IL-33, and IgG levels, as a way of predicting who's going to get on better on Rituxan.
It's a strong effect. That's something you can talk about with the patients. Sometimes even simple things. So this is an analysis from Japan looking at changes in white blood cells after tocilizumab therapy and a change in eosinophils. And actually an interesting effect that based upon how their white blood cell counts and the differential, you may have response or non-response to a particular drug. So I think what we'll see is a pattern of putting things together to see, can we get something? And of course the bar is high. We have to have a very strong predictor before we're able to use it as something we talk to patients about.
So let's wrap up with the last two slides. Let's do this one. what's your take on zoster and this review?
So this was interesting because isn't this something we wrestle with all the time? Yesterday in clinic, I told a patient, yes, they brought it up, and I felt bad that I didn't bring it up. I said, yes, they should get the shingles vaccine, but they were 40. And I said, it's going to be an issue. You may not get it covered. This is a patient with psoriatic arthritis, actually. I said, it's a good idea. I can't do it. We don't stock vaccines in our office. You have to go to the internist. The internist might say, no, you're 40, get out of here. But if you look at this, if you accept that the idea of vaccinating people who are over 60 makes sense, you look at the risk of zoster per 1,000 patient years. What if you said, well, if that's acceptable, who among our other diseases should be vaccinated? And that's in red. You see, lots of people should. So we should be vaccinating early. If it makes sense for a healthy 60-year-old without one of our diseases, it absolutely makes sense. In some ways, a lot more sense for our younger patients who have this. And it's just, we're not doing it. I don't know.
And it's not paid for by payers and not actually, not even advised by the CDC until age 60. And they've looked at younger groups and showed actually better responses in some cases. So this needs to be liberalized, but we actually need better ways of vaccinating patients. Let's end with, these are the ACR guidelines, which I don't think anybody can read this slide because it's, or maybe nobody can interpret the guidelines. Well, yeah. That might be another.
Well, it's interesting. Who was ever at the ACR meeting, they put it in a room that was kind of an okay-sized room that was just filled to the gills, as if they knew it was an incredibly important topic, but they wanted to select only those people who really, really wanted to be, which is mostly investors in pharma. That's who was in the audience. But it'd be interesting. I think it was led by Jazz Singh from UAB. I think he tried to do a good job. I think personally, sometimes guidelines get caught up in the weeds of the process of doing it. So now there's the McMaster group has a whole cottage industry about how you're supposed to do guidelines called GRADE. And sometimes that the following the process interferes with actually doing this. But we'll see. These will be out, I think, relatively soon. I think a lot of it will be mom and apple pie, everybody will agree with. Some of it will be a little bit contentious, particularly those areas where you stray away from solid evidence that tells us this is a good idea and get into people's opinions and biases. For example, treating patients who had a malignancy, which is another big clinical problem that we don't have good data for.
So there's instructional value to these, certainly. I think one of the mistakes of these guidelines were that they, number one, did away with the importance of the prognostic risk factors. Those are no longer in the decision-making process. And I think another major mistake was in how they handle cancer. They continued to be afraid of the early data that came out about TNF inhibitors in lymphoma and cancer, when right now, if you have a patient who has cancer, what's the drug, or a history of cancer, or a worry of cancer, what's the drug they should go on? A TNF inhibitor, because there's a ton of evidence about what's happened to those people. The drugs they recommend are the drugs that actually there is no evidence, rituximab and all the other choices that they recommend. So the guideline group, I think, missed that whole point about the available evidence that's out there. But nonetheless, these are things that we will debate quite a bit going forward.